covalent drug has been always just started. Covalent drug has drawn quite a lot of attention, especially recently, due to the COVID main protease inhibitor design. So those kind of drugs, they carry an electrophilic warhead. So when the ligand binds to the enzyme catalytic site, the warhead will form covalent bond with a catalytic residue. In this case, is cysteine. And because the warhead is relatively weak, so this covalent bond can actually break. So the covalent bond state can actually go back to the non-covalent bond state. And because both steps are reversible, so we can actually take advantage of the thermodynamic equilibrium, and we can optimize those reversible covalent drugs by either modifying the electrophilicity of warheads. So this has been done for quite a long time. Nowadays, you can find a whole library of warhead with different electrophilicity you can choose. A second approach is relatively new, and what I mean is by besides this small warhead, you have this large, the rest part of your large molecule that you can use to fine-tune the non-covalent interaction. And this is especially useful for a relatively weak warhead. So your binding fringe difference is not too large. So the hope is, as you see, the reversible covalent drug can be more potent and has longer residence time than the conventional non-covalent drugs, but it can also be more selective, so less toxic than the conventional irreversible covalent drugs, right? So that's the hope, but Professor Jorgensen just proved us there are exceptions, which is very impressive. So I came to know this kind of ligand actually, okay. Already 7 years ago, I have a colleague, Michel Baudry, he's a neuroscientist, and he discovered that in our brain, we have two kinds of calcium-dependent cysteine protease, calpin-1 and calpin-2, and they have opposite effects on our neurons. So calpin-1 is a brake of neuron degeneration, while calpin-2 is an accelerator. So obviously, you will realize the magic bullet to protect our neuron has to be the inhibitor that only inhibit calpin-2, because if you inhibit both, you have no effect on a neuron. So when I overlay these two isoform protein, I found that, so the blue is conserved region, red is non-conserved. I found that the catalytic site is very conserved, same as all other proteins. But there was one site which is at the P1 prime site next to the warhead, which is also the S1 prime site on the enzyme, that has some difference between the two enzymes. So we wanted to modify this site to increase the calpin-2 selectivity while keeping the ketoamine warhead. Actually, ketoamine warhead is one of the rare warheads that you can actually extend to the P1 prime site. So we want to keep it. So as I said, this project started 7 years ago. At that time, there was no FEP application on this kind of ligand. So my first question was just, whether I can apply FEP on this kind of ligand. So if you look at the simple diagram with two states, so the overall KD should depend on, the concentration of the total products over the concentration of the reactants. And in this case, you have two different kind of products. So you have a non-covalent bonding state and covalent binding state. So then the total delta G will depend on the delta G of covalent states. Here I call delta G DC, from dissociation to covalent. And also the delta G of DM, which is from dissociation to the non-covalent bound state. So of course, if one of the delta G dominant, right, more negative, then the second term is negligible. But that's exactly what I wanted to find out, whether we can just use one term to predict. And I'll show you a result actually was quite interesting, that we show even you keep the same warhead, but just by modifying the non-covalent interaction, the relation between these two delta G actually changes. So this delta MC, its difference between these two actually changes a lot. And so because our project is only to modify the non-covalent interaction of the R group without affecting the electrophilicity of the warhead, we can split this 2 delta G into 4 terms. So in the case, in the non-covalent state, you can compute the absolute binding free energy of a reference compound in the covalent bonded state, and then in the non-covalent bonded state. And then the rest of the compound, you can compute the relative binding free energy in the covalent state and non-covalent state. So all these 3 terms are easy to compute, right? The problem is this covalent bonded state, it involves, bond breaking and forming. So that's a bottleneck for covalent drug design, actually. But people has done that using QMMM. It's possible. But if you just want to rank your compound without, with some estimation, there's also a trick you can do is you can compute the total delta G from the KD of your reference compound, and then you can just subtract this non-covalent term to estimate the covalent term. So this is quickly jump to our results. So we have the 3 delta G. So we found that for our limited number of compounds, the covalent binding free energy is higher. Most of them is more than 3 kcal higher, 3 to 10 kcal higher than the non-covalent binding state. So the total delta G is almost identical as the covalent state. That's good news. Although there's one compound which we replace the benzyl group with the S-ethanol group, we found that the covalent state binding free energy decrease and non-covalent increase. In this case, the total delta G is actually the sum of the 2 exponential terms. So if you plot, the correlation with the experimental delta G, obviously the non-covalent state has no correlation. So that's actually the take-home message. We learned that we should not perform FEP using non-covalent state. Actually, in some cases, we even see anti-correlation. And we have some wild speculation on this paper in the discussion section. So it works to, really parameterize this covalent bond, and then you do the FEP using the covalent state. But also there's exception, as we show, one of the compound actually changes the relation between the 2 delta G. So that's basically a proof of concept and benchmark of how do we do FEP for those covalent ligand. But FEP, the problem is it does not tell us how to design the ligand. So for that, we actually relying on this Silks method, which has been introduced in yesterday's poster section. So basically, we run 10 copy of the 100 nanosecond in the solution, and then you map the density of those fragments. So it gives us very good idea, where for calpin 2, we need a non-polar group at meta position. Calpin 1 need a non-polar group in para position. We later synthesized compound, we found that bulky group of the para position increased the calpin 1 selectivity by 100-fold. The water map was also very useful. Silks provide the water map. We found the water density at this P1 prime location only in calpin 2, not in calpin 1. And we introduced the hydroxyl group OH as a position and increased the calpin 2 selectivity by actually 4-fold. So that was quite useful. Unfortunately, when we introduced the OH group, we also introduced the additional chiral center. So that idea was killed by our medicinal chemist. I'm sure you have a similar experience. So the fragment map can also be used for docking, but the Silks docking was originally, designed for non-covalent binding. So we actually also tested, come up with a protocol that you can use to do the fragment docking for covalent ligands. So now we have a workflow is, we have a Silk map to help us design. So we actually have a delta map. We subtract the calpin 2 over from the calpin 1 map. So we have a delta map just to target selectivity. And then we use our fragment docking protocol published in this paper for the initial screening, and then using the covalent FEP for the final screening. So that's about it. A second part is allosteric agonist. So allosteric agonist is probably the most tricky drug to design. The reason is because the ligand has to bind to the receptor, the protein, but then it's not enough. It also has to trigger a conformational change in a remote site. So that cannot be only predicted by a single binding affinity. So, and the target, how do I remove this? So the target we have is also quite tricky. It's mechanosensitive piezo channel. So among all the ion channels in our body, piezo is the one that's most sensitive to force. So piezo is the reason that we can sense very soft touch on our skin. It's also the reason that you feel full after you eat and you know when you need to go. Okay, so that's how important it is. So the group in Scripps who discovered this protein, they also did a high throughput screening using Novartis compound library. And they found this small molecule, just this one small molecule that can activate piezo-1. So they call it UDA1 because it can basically manipulate the force sensor, which is a piezo on our body. The problem is nobody knows where this small molecule will bind to the piezo, so that I haven't mentioned this piezo channel is giant. It has more than 8,000 amino acids. It's a trimer, so it has 3 long arms that curve up, actually binds the band of the cell membrane, make it a dome shape, inverse dome shape. This is a top view. You can see this curved arm. It looks a lot like the Sicilian flag. In the middle is the channel pore. It is closed in all the solved cryo-EM structures. So my lab take upon this challenge, and we wanted to find the binding site. Since nobody knows where those molecules should go, we choose to perform saturated binding. So we put 20 UDA molecule in the solution. They quickly partition into the membrane, and during the 4.8 microseconds, they were able to sample, all over the transmembrane region. And then we plot the mobility of each ligand, which is just a windowed IMSF. So the blue means more stable, red is not stable. And we found the 2 candidates. The ligand 13 was stable for 3 microseconds. The second one is the ligand 20. It was less stable, but still stayed in a binding pocket for 4 microseconds. So our experimental collaborator, Jerome Lacroix, was able to conduct a tryptophan scanning. So he was able to mutate small alanine to tryptophan, the bulky side chain, to block the binding site. And he was able to rule out the ligand 20 binding site and confirm the ligand 13 binding site. So where is this ligand 13 bind on this giant protein? Let me show you. Okay. Here it is. So this UDA molecule actually binds at the arm, at the elbow of the arm region, which is 40 angstrom away from the pore. So we were very puzzled. How can this small ligand, trigger the pore opening that's 40 angstrom away? And so there was a very small SAR study. They show that if they change the position of the N, those analog behave similar to UDA but weaker, so like partial agonist. But there was just one molecule, which is that they replaced this pyrazine by pyrrole, which has a hydrogen bond donor, and sulfur to oxygen. And this molecule had no effect on piezo, but when they apply together, put UDA and DOCU both in the system, they found that this molecule inhibits UDA's activity on piezo. So they name it as DOCU for obvious reason. And so we want to see, the structure is quite similar. So we want to see, where the difference came from. Right, so we have 2 options, right? You can do relative binding free energy or absolute binding free energy. Relative, you have the 2 range difference. So now we decide to conduct absolute binding free energy because we also don't want to assume that the binding poles are identical overlay. So that was fortunate, because if we decide to use a relative binding free energy, we will completely miss the mechanism. And because this channel is huge, so we pick just one arm, so one subunit out of the whole trimer to conduct FEP, since we know that FEP mostly rely on the local conformational change. So this is the FEP Hamiltonian rapid exchange region, and this is zoom in to see where the small molecule binds. So the only difference between here compared with rest of the FEP is perhaps our ligand has to first permeate from the solution into the membrane, and then from membrane binds to the protein. But since this is in the membrane permeation is an intermediate step, we actually don't have to calculate the log P explicitly, so we can still use the protein, decouple the ligand from the protein and from the solution to compute the DRG. What we got is that the DOCU binds similar binding site, slightly shift, actually binds to the piezo stronger than UDA. Their solvation for energy is almost identical. So that does not explain us why UDA is agonist, DOCU is does not do anything on piezo, right? Actually binds stronger. So to do that, to investigate, we actually have to we have to compute the FEP in piezo open state. As I said, there was no open state structure available. So my lab actually generated an open state using a crowding-induced membrane flattening. So it's actually a trick that, because I said the piezo generates a dome-shaped membrane, and if you have the two image closer, then the bending free energy of the membrane will actually flatten the piezo arm, and that induce the pore opening. And of course, we have to do tons of tests for conductance measurement, mutation, ionic selectivity, just to show that it is a reasonable open state, agree with all the experimental data, not just artificially stretched pore. So here I'm only interested in the UDA binding site. So you can see that from orange to cyan, close to open, there was one helix tilted up. And the result is that the bottom of the binding site, so that's the bottom of the helix bundle, opened up in open state. And the UDA actually binds deeper in the open state because there's more space. So now we can compute the binding for energy to open state also. And we found that UDA and DOCU actually binds to the open state equally strong and similar as the DOCU in closed state. So the only difference is actually UDA binds weaker in the closed piezo state. That's the only difference. And so I have people ask me, so does UDA acting as acting through conformational selection? So I said, well, actually, it doesn't matter if it's conformation selection or induced fit. So I draw this diagram to show that right now we know that UDA binds to the open state more favorable. So PO is piezo open, PC is piezo closed. So DRG2 is more negative than DRG1. So that's a conformational selection. But because the thermodynamic cycle has to sum up to 0, it also means DRG4 is smaller than DRG3. So this means when piezo bound to UDA, it's easier to open compared with when piezo has no UDA. So that basically tells you it is an agonist, and either through conformational selection or induction, it basically favors the open state by 2.8 kcal per mole, while DOCU has no difference. That's why it is a silent binder, although it binds quite strong. And so to facilitate the further design, of course, we need to look at the structure. So structure-wise, we see that, so I said UDA binds deeper into the binding pocket, because the binding pocket opened up. However, DOCU, because DOCU actually has a hydrogen bond donor, while if you look at UDA, it has no hydrogen bond donor at all. And we see that it formed a hydrogen bond with a proline backbone, and that prevented DOCU to bind deeper down. So the binding pose is quite different. And so that was that's actually agree with our wedge mechanism for UDA on piezo. We previously did a membrane stretching simulation, and we proposed that when UDA binds to the piezo arm, and when arm open up, UDA can bind even deeper. So that acting as a wedge to prevent arm closing. And this mechanism was also consistent with this small SAR study showing that most of the compound that does not have a hydrogen bond donor, they have partial agonist effect. So last is, so now we know how to differentiate agonist from silent binder. But if we want to use that to do lead optimization for each compound, we have to run 2 absolute binding free energy in open state and closed state. So that's still quite expensive. So what we want is have a faster way to differentiate agonist from silent binder. And we started to use this allosteric network topology clustering. So when UDA binds 14 angstrom away from the pore, so you basically, the binding site can serve as source and the pore is a sink. So between source and sink, you can map up an allosteric pathway. The problem is a conventional way is the allosteric pathway is generated using pairwise correlation. So either Pearson correlation, mutual information, or residue pairwise interaction. But you see those pairwise score, they have no information on source and sink. And the problem is when you run a 4 replica simulation, you will get 4 different network topologies, so that you cannot really use that for prediction. So my postdoc Wesley came up with this current flow bittriness. So you can compute the bittriness between the amino acid with between the source and sink nodes. So it's the same concept as you compute the electric current flow through an electron network using Kirchhoff's law. And you can see that the network became very focused between source and sink. And if you run 4 replica, your topology is also very consistent. So we tested that. We run 6 replicas of short simulations, so much easier than, much faster than FEP. We did an 8-pole, and then we did Doku and UDA, and use a simple clustering method. We already see that they can be separated in the high dimensional space. So right now we have more compounds available. So we're planning to, so we can learn how to cluster, better clustering methods. And we're looking for actually a collaborator to help us with this project. But the network code is available at GitHub, and there's also a tutorial on this book chapter. Last but not least, all credit goes to my group members. So Wesley, Wenjuan, Yichun, Bella, and Payo. They did all the work, and collaborator Jerome Lacroix for Piezo project, Michelle Beaudry for the Kelvin project. And I also want to mention that besides our chemical approach, we also compute KD and KL from the Markovian milestoning. So on this paper, you can see that's also a different kind of binding study. And yeah, that's it. Thanks.
Thanks, Lyna, for a great talk. So again, I'm clapping on behalf of the audience, but thanks for your presentation. So I have a question from the audience is, aren't your R groups too close to the warhead, to the point that you would consider them as different warheads since they might affect the electrophilicity of the warhead?
Yeah, great question. So we actually discussed that in our paper. So first of all, you want to make sure that it's not conjugated, right? If your R group is conjugated with warhead, then that totally give you a different warhead. So in our case, we do have a CH3 bridging all the other R groups, so they are not directly connected to the R group. In that case, you don't have the resonance or induction effect, so the electrophilicity can be maintained. But yeah, of course, there's no guarantee. So that's actually one of the caveats we discuss in the paper also. Thank you.
And then one question for me, I'm not very familiar with reversible covalent inhibitors, but are there cases in which the 2-state model that you have might not work so well? For example, if you have an intermediate step where you need protein reorganization or something like that.
Yeah, definitely. So this is very simplified So we actually discussed in the GCIM paper also that it also depends on the kinetics of your reversible covalent binding, because you know the whole thing we are drawing, this thermodynamic cycle, we assume the reaction reaches thermodynamic cycle, but it's not necessarily true. For some covalent bonds, you know, you might never have time to reach the intermediate step. And also for some reaction, maybe the proton transfer is a rate limiting step. So those are missing in the simple two-state model. But it is so what we show, I think the most important message is that the non-covalent state should not be used to rank covalent you know, drug. And for in terms of ranking purpose, you know, the covalent state FEP is actually quite useful. That's at least what I learned.
Okay, thank you very much. So that brings us to the end of our meeting. So thanks again to all the speakers and panelists who spoke today, and our organizers that have been working here on the background, and of course our sponsors. And also thank everyone for staying a little bit later than the meeting planned time. But that's it for today, and I will see you all tomorrow.